Hello students, welcome to my lecture. In the previous lecture, I had explained you the sexual reproduction in flowering plant, especially the part of the male reproductive structure. In this lecture, I will explain you the female reproductive structure in the flowering plant. So let's start with the content. Okay, the female reproductive structure of the flower is known as gynoecium. This gynoecium is also known as pistil and carpel. So these three terms are representing the same thing, that is the female reproductive part of the flower. So if the flower is having only single pistil, single female reproductive part, then it is known as monocarpellary. It means it is having only one carpel. So this is monocarpellary. But if the flower is having more than one pistil, then it is known as multicarpellary. So it depends on the number of pistil, number of carpel present inside the flower, right? So, but if the flower is having more than one pistil, then two conditions are there. Condition number one, okay, these pistils may be fused. So if the fused, then it is syncarpous condition. But if the pistils are free, then this condition is known as apocarpous condition. So there are two conditions: pistils are fused, syncarpous condition, and pistils are free, apocarpous condition. Now move ahead. Each pistil has three different parts. These parts are stigma, style, and ovary. So in this slide you can see, okay, this is the stigma part. This is a long tube style part, and at the base it is ovary. Stigma act as a landing platform for the pollen grain. It means it is the receptor site of the pollen grain attached on this stigma part. Now these pollen grain travel through the elongated cylindrical part, that is the style, and finally they reaches at the basal bulged part, that is known as ovary. So stigma, receiving of pollen grain; style, traveling of the pollen grain; ovary, the final destination of the pollen grain. So in this slide you can see this is the stigma, here is the style, and at the base this is the ovary. Now move ahead. Inside the ovary you will find the cavity. This cavity is known as ovarian cavity. It is also known as locule. So what is locule? Locule is the cavity which present inside the ovary, and the placenta, which is well connected with the ovarian cavity. So in this diagram you can see, here is the placenta. This is the site of placenta, and this placenta connected with the locule, that is the ovarian cavity. Now ovule is arising from the placenta. So you can see here is the placenta, and this complete structure, this is the ovule. So this ovule arising from the placenta, this ovule is also known as megasporogenesis. क्या कहलाता है? It is also known as megasporogenesis. Now the number of ovule, the number of megasporogenesis vary from plant to plant, flower to flower, right? So if the flower is having only one ovule, then we can identify these plants by the presence of seed inside the fruit. Means if only one seed is present inside the fruit, then we can say only one ovule is present inside the ovary. But if the more than one seed are present inside the fruit, then we can say this ovary having more than one ovule. So wheat, paddy, rice, mango, they are having only one ovule inside the ovary. But some other plant like papaya, watermelon, muskmelon, orchids, guava, they are having more than one ovule inside the ovary. Move ahead with the next part. Now, the structure of the ovule. In this slide, you can see the structure of ovule. It is having the different regions. Ovule attached to the placenta. So I told you this is the placenta. Now ovule is attached to the placenta with the funicle, the stalk, the handle-like structure. So here is the funicle. This is the funicle. Now one more point, that is hilum. So hilum is the junction between the funicle and the ovule. So here this is the hilum. This is the hilum because यहाँ तक क्या है funicle. So this part is the funicle. Now the junction between the funicle and the complete ovule, this is the complete ovule, this is known as hilum. Now ovule has various, generally one or two protective envelopes. These are known as integument, the outer integument and the inner integument. The purpose of the integument is to protect the developing embryo sac, right? Now, these integuments are present all over the embryo sac, but at this point, at this aperture, these integuments are absent. This and this point is known as micropyle. What we call it? Micropyle. So ovule has two ends. One is the chalazal end, the base, and another is the micropylar end. So it is having the micropylar end and chalazal end. In this diagram, you can see that this is the ovary wall, right? This is the ovary wall, and here is the placenta. Now, this placenta, as I told you, joined with the funicle. So this diagram is showing the two ovule per ovary, right? So this is the multi-carpellary condition. So two ovule per ovary. Now, these are the two ovules which present inside the ovary, and this complete space is known as ovarian cavity. So move ahead with this part. Now this slide is showing the enlarged view of the ovule. In this diagram, you can very well see the position of the funicle, hilum, the integument, outer one, inner one. Here is the embryo sac, and this is the nucellus area. This nucellus present beneath the integuments. So this is a mass of cell which contain reserve food material, and this food material is required for the development of the embryo sac. So inside the nucellus there is an embryo sac which is known as female gametophyte. So this is the embryo sac. It is also known as female gametophyte. In general, single ovule have only one embryo sac. This is a general phenomena. So this is the complete ovule, and this is having only one embryo sac. This embryo sac is formed from the megaspore. Now I will tell you the process of the formation of the megaspore. The process of formation of the embryo sac from the megaspore. This process is known as megasporogenesis. So what exactly megasporogenesis? It is the process of the formation of the megaspore from the megaspore mother cell. So ये क्या है? Megaspore mother cell से जो finally embryo sac बन रहा है, megaspore बन रहा है, that process is known as megasporogenesis. So ovule generally differentiate Here is the division. Differentiate a single megaspore mother cell in the micropylar region of the nucellus. तो देखिए इस स्लाइड में देखिएगा कि ये जो पार्ट है it is at the micropylar end, right? Now this single megaspore, large, having dense cytoplasm, undergo the meiosis and it will form 4 megaspore. So this is the single megaspore mother cell. Through the successive stages after the meiosis, 1, 2, 3, 4, 4 megaspore, and this condition is known as megaspore tetrad condition. Tetrad means 4. Tetra word means 4. Now this diagram is showing the 4 megaspores, the megaspore tetrad. Now out of the 4 megaspores, 3 megaspore degenerate, 3 खत्म हो जाते हैं, right? Only one functional megaspore remains there which develop into the female gametophyte. तो अभी यहाँ पे 4 थे, 1 2 3 4, इसमें से 3 खराब हो गए, degenerate हो गए, एक functional megaspore बचा. Now this embryo sac which is formed from a single megaspore is known as monosporic development. It means जो एक embryo sac इससे बनेगा वो कहलाएगा आपका monosporic development. चलिए आगे बात करते हैं. Now formation of the female gametophyte embryo sac. Now nucleus, that is the functional megaspore, first it divide mitotically. The mitosis means two opposite nuclei are there. Now it will again undergo one more mitosis, then it form 2 plus 2, total 4 nuclei. It will again undergo one more mitosis, then it will form total 4 plus 4, 8 nuclei. Now this condition is known as 8 nucleate stage of the embryo sac. So out of the 8 nuclei, 3 nuclei moves towards the micropylar end, 3 moves towards the chalazal end, and 2 remain present at the center of the embryo sac. Move ahead. So the 3 nuclei that present at the micropylar end, they form an egg, and 2 are forming synergid, right? क्या बना रहे हैं? Synergid. These synergid also have a special structure, that is the filiform apparatus, which provide the direction and guidelines for the pollen grain to reach towards the egg. 3 antipodal cells which present at the chalazal end, they also degenerate, and the 2 cells that present at the center, they fused together, and this is known as the large central cell. Now look at this diagram. This is the condition, the 8 nucleate and 7 celled stage, which means 1 2 3 4 5 6 7 8. Total nuclei are 8, but the cell 1 2 3 4 5 6, and this is the combined cell, the 7 cell. So this is known as 7 celled 8 nucleate stage, right? Now this is the complete diagram which is showing the female gametophyte. So in this slide you can very well understand the overall functioning, overall formation of the female gametophyte. This is the megaspore undergo meiosis, 3 degenerate, 1 functional, mitosis, one more mitosis, 3 at the micropyle end, 3 at the chalazal end, 2 at the center. So these 2 central cells will form the endosperm inside the ovule, right? So out of the 3, 2 are the synergid, 1 will act, and these three antipodal will degenerate after some time. So this is all